Okay, so in this video we talk about lipase and lipoprotein metabolism. Lipase and metabolism. So it's very important as a medical lab science to know what goes through lipase and lipoprotein metabolism, because in the lab one of the common tests that we do perform is lipid profile. So you'll be asked to check the levels of cholesterol, maybe total cholesterol, maybe high let's say triglyceride or triglyceride, to low density lipoprotein. All these things are what we do. So why do we do those tests? You understand? What is the brain behind doing those tests? It is very, very important as a laboratory scientist to know the role of lipids in the body. So today I want us to go through lipase and lipoprotein metabolism. Lipase and lipoprotein metabolism. Very, very important, and I think at the end of this lesson we will be able to know what goes through lipase metabolism in the body. Okay, so objectives: 1, I'll talk about the definition. I'll also throw more light on the importance of lipids. Okay, why are lipids important? So I'll also talk about lipids transport and metabolism in the body. Then I will possibly end by looking at disorders of lipid metabolism. Okay, so a whole lot of topics. Let's continue and deal with it. So definition: what are lipids? So we say lipids or lipid is any of various organic compounds that are insoluble in water. Okay, when we say insoluble in water, it means it is not water loving. One term that science we use for things that are not water loving, we call them hydrophobic. Okay, hydrophobic. It means that lipids are hydrophobic. They cannot dissolve in water. And so of course it is true, because if anything is hydrophobic, you don't expect that thing to dissolve in water. Very important. So put that in mind, that lipids are organic compounds that are insoluble in water. Okay, so anywhere in the body where there is water, for instance, let's take the blood. The blood is about 95% plasma. Blood is about plasma and cells. Okay, and the plasma is about 95% water. And so lipids cannot dissolve in the aqueous layer or aqueous component, that is the plasma portion of blood, because they are hydrophobic, cannot dissolve in water. So what I want you to take it from here is that lipids cannot dissolve in water. They are insoluble in water. Another point that you have to know is that lipids, we have different types of lipids. Okay, so we have fatty acid containing lipids, those that contain fatty acid. Okay, example, we have the wax, we have the phospholipids, we have the sphingolipid, and the fat and oil. Okay, these lipids contain fatty acid. Okay, and then steroid is a lipid that is a non-fatty acid containing lipids. So bro, we can group lipids into fatty acid containing lipids and non-fatty acid containing lipids. Very important to know. So what are some of the importance of lipids? Very, very, very important. So cholesterol and phospholipids are vital structural components of cellular membrane. Okay, so when you take these two lipids, cholesterol and phospholipids, they are found on the cell membrane, so they are structural components of cell membrane. So some lipids are vital structural components of cellular membrane, which is very important. Okay, very important. Now you can also say cholesterol is the precursor of steroid hormones and bile acid. Okay, so before we can synthesize or make steroid hormones in the body, you have to get cholesterol, which serves as a precursor. Okay, so example of steroid hormone, we can talk about the estrogen, testosterone, and those progesterone, those stuff, right? So it becomes important that if you have cholesterol, it serves as a precursor for those hormones. So one importance of lipids is that Cholesterol being lipids serve as a precursor for steroid hormones and bile acid. Okay. Now triglyceride is an important storage and transport form of energy. Now we take triglyceride. It is an important storage and transport form of energy. Okay, very very important. Now absorption of dietary lipid is essential for absorption of fat soluble vitamins. Okay, so lipids also help in the absorption of some fat soluble vitamins. Okay, I made mention of some of them, K and D, the rest. So very very important. So these are some importance of lipids. So lipid digestion, how does digestion of lipids occur? So this diagram tests it, or so what I would say is that lipids end product is fatty acid and glycerol, right? Good. So how does the digestion of lipids occur? Now in the mouth there is an enzyme we call the salivary lipase. Salivary lipase. It digests lipids in the mouth. Okay, it digests lipids in the mouth. Now what happens is that in the mouth, when you take in any lipids food, digestion in the mouth brings it straight to the heart, the duodenum, because back to digestion, you know that when you take the alimentary canal, digestion of lipids one occurs in the mouth and the duodenum, then it comes to the small intestine, because the stomach, there are not enzymes that digest lipids there. Okay, so in the mouth, okay, in the mouth, this is the esophagus linking the mouth to the stomach. Okay, now from the mouth, the lipids food, okay, come down to the duodenum, this portion. Now in the duodenum, that is where most of the activities occur. In the duodenum, what happens is that there are chemoreceptors, okay, chemoreceptors that are found in the duodenum. Okay, now upon arrival of lipids in the duodenum, what the chemoreceptors do is that they will stimulate the brain. They are nerves that connect the duodenum to the brain. Okay, and these chemoreceptors stimulate the brain. Okay, that brain, there are fatty acid in the duodenum, so they must be hot break down or broken down. So what the brain also do is that it will sense another hormone we call secretin. Okay, secretin. Now what secretin does is that it causes the release, this is the pancreas. Okay, can you see that? Causes the release of an enzyme we call the pancreatic lipase. Pancreatic lipase. And then this pancreatic lipase, what it does is that it comes into the duodenum. Okay, released in the pancreas, it comes into the duodenum to break down the lipids in the duodenum. Okay, now there is a cofactor to this pancreatic lipase we call the colipase. Okay, it helps the pancreatic lipase to break down the lipids. Okay, so folks, what I'm trying to say is that in the duodenum, when lipids arrive in the duodenum, there must be release of pancreatic lipase by the hot pancreas. Okay, but then before it happens, chemoreceptors in the duodenum, okay, stimulate the brain to sense the secretin. Secretin is basically a hormone that stimulates the release of pancreatic lipase. So the moment there is secretin activation, pancreatic lipase will cause the release of, or pancreatic lipase will be released from the pancreas. Okay, so from the pancreas, the pancreatic lipase will be released into the duodenum. Then it will ensure the breakdown of these lipids. Okay, now when these lipids are broken down, they move straight to the small intestine. Okay, small intestine. So in the lumen of the small intestine, can you see this? There are digested lipids there, and those digested lipids are called micelle. Okay, micelle, micelle, micelle. Now this micelle a combination of lipids can get the triacylglycerol, the monoacylglycerol. We can get the bar salts, we can also get the cholesterol and a whole lot of them. Okay, so mast cells are a group of or a combination of triglyceride, cholesterol, monoacylglyceride, okay, that are being broken down by the pancreatic lipase. You understand that? So now these mast cells are in the lumen of the small intestine, but they shouldn't be there. They must go to the liver so that they can enter the bloodstream for the various tissue to use them up. Okay, so how does the mast cells get into the bloodstream? Okay, very important, very important. So this lipase, okay, this mast cell, which I said the comprises of the triacylglycerol, the monoacylglycerol, the cholesterol, and the bile salts. In the small intestine, the product from fat digestion and bile acid form a mast cell, like I said, which moves towards the microvilli to allow the lipids to diffuse into the mucosal cells. Okay, so inside the mucosal cells, the fatty acid and monoglyceride are reassembled into triglyceride and incorporated into lipids. Now, lipid transport particles called chiromicrons, which enter the lymph vessel. Okay, very important. So inside the mucosa, this is the mucosa of the small intestine. What happens there? The fatty acid and the monoacylglyceride, like the mast cell, okay, they are reassembled into triglyceride, okay, and then incorporated into lipid transport particles. So the lipid transport particles there is called the chiromicrons. Then they enter the lymphatic vessel. Now in the lymphatic vessel, there are so many things that we will talk about that when we get there. Okay, so what I want you to take from the digestion of the lipids is that from the mouth to the duodenum. In the duodenum, they must be broken down by an enzyme called the pancreatic lipase, which is released by the pancreas. After the pancreatic lipase has performed its function, we are going to get mast cell, which consists of triacylglyceride, monoacylglyceride, diacylglyceride, cholesterol, and those stuff. Okay, now they are found in the lumen of the small intestine. Okay, and this mast cell, okay, moves straight to the heart, the liver. Okay, now how do they move to the liver? Now the mast cells are first absorbed into the enterocyte. Basically, the enterocyte is a epithelial cell lining the lumen of the intestine. Okay, so there's an epithelial cell lining the lumen of the small intestine called enterocyte. Now the mast cells in the enterocyte migrate to the lactea. So the lactea are the lymphatic vessel or the lymphatic duct that lines the GIT. Okay, so from the enterocyte to the lactea, and they move through straight to the cystine chili. From the cystine chili, then they go to the jugular duct, then they move to the thoracic, and finally they enter the liver. Okay, so what I want you to take from here is that the mast cells are finally getting to the liver. What is going to happen in the liver? Very important. There's going to be what is called formation of lipoproteins. What are the roles of lipoproteins? We're going to talk about them in no time. Okay, so this is the digestion process of lipids. Very important. So lipoproteins and apolipoproteins, we talk about them one after the other. So lipoproteins, let's talk about lipoproteins. So lipoproteins basically their formation. So the formation of lipoproteins by the mast cells occurs in the liver, like I was explaining. So lipoproteins are complex spherical structure. They have complex spherical structure that have a hydrophobic core wrapped in a hydrophilic coating. Okay, so what happens is that when you take lipoproteins, okay, there are two coating. There are two cores, the inner core and the outer core. The inner core, okay, is hydrophobic. The inner core is hydrophobic, like it does not dissolve in water, okay, and the outer core is hydrophilic. So take note of that. That lipoproteins, you have the inner core and the outer core. The inner core hydrophobic, the outer core is hydrophilic. Now the core contains triglyceride and cholesterol esters. That is the inner core. The portion which is hydrophobic contains triglyceride and cholesterol ester, while the surface contains the phospholipase, free cholesterol and proteins. Most of the times they are apolipoproteins, okay. So if in exam they ask you to mention the composition of the inner core of a lipoprotein, it is going to be triglyceride and cholesterol ester, whilst the surface we have the phospholipase, free cholesterol and proteins. Most of the times the apolipoproteins, okay, very important. So lipids are generally hydrophobic and therefore insoluble in water. This point we have established it long time. So only the charged phosphate groups of phospholipid and hydroxyl group of free cholesterol are hydrophobic. So it means that lipids cannot be transported in plasma, so they need something to transport them, okay. But here we are saying that for the lipoproteins, the outer core consists of phospholipids and free cholesterol, and I'm trying to tell you that when you take the lipoproteins, the outer core is hydrophilic, okay, because the free cholesterol contains hydroxy group, and the phosphate group on the phospholipid also is hydrophilic. So that lipoproteins appear to solve the problem of transporting lipids in plasma. So for lipids to be transported in plasma, okay, the lipids are transported in plasma as lipoprotein, because these lipoproteins are hydrophilic. Very important. So lipoproteins, we have about 5 of them, okay. We have the chylomicron. The first one is called chylomicron. The second one is very low density lipoprotein. Very low density lipoprotein. The next one is intermediate lipoprotein. Okay, intermediate lipoprotein. And now we have the low and high density lipoprotein. Very important. So about 5 of them.
I'd love to help, but you'll need to update Windows first. You can do that by using Windows Update Assistant.
So that is that. We have the chylomicron, the VLDL, the IDL, LDL, and the HDL. Okay, these are the 5 lipoproteins that we have. Very important. So there is a Asberg C model of plasma lipoprotein. How does lipoprotein look like? So this is the C model, okay. So the inner core, can you see that? See where this arrow is pointing? This is the inner core. Another one is this. So the inner core consists of cholesterol esters and triglyceride, okay. Very important. The inner core, cholesterol esters and triglyceride. And the outer core, we can have the phospholipid, free cholesterol, and the apolipoproteins, okay. Remember I told you that the inner core are hydrophobic, while the outer core are hydrophilic. Why? Because of the hydroxy group of the free cholesterol and the phosphate group of the phospholipid. Very important. So lipoproteins vary in size, density, and electrophoretic mobility. That one is very important. And the greater the triglyceride content, the larger and less dense the lipoprotein particle is, okay. So every lipoprotein has a certain amount of triglyceride content, So if you take chylomicron, okay, chylomicron, it has about 90 triglyceride content, okay, which makes the triglyceride content in chylomicron very hot, large. And because of that great amount of triglyceride content in chylomicron, it becomes less dense, because the larger the triglyceride content, the less dense the lipoprotein is, okay. Now the chylomicrons are the most triglyceride rich, and then chylomicron is the main carrier of dietary triglyceride, okay. So the main carrier of dietary triglyceride, they are the chylomicron, very important. They transport the triglyceride that we eat from our diet in the body. And the VLDL, very low density lipoprotein, is the main carrier of endogenous triglyceride. Endogenous triglyceride, okay. So endogenous triglyceride, they are being carried by the VLDL. Now low density lipoprotein is the main carrier of cholesterol, okay. The cholesterol content is very high, so we call it bad cholesterol. The LDL, bad cholesterol, is the main carrier of cholesterol. And then the HDL carries cholesterol from non-hepatic tissue. That one is called good cholesterol. So if you look at this diagram, we have the lipoprotein, the density, the electrophoretic mobility, we have the triglyceride content, and apolipoproteins bound to that lipoprotein. So if you take the chylomicron, the density is less than 0.96. In electrophoretic mobility, it still remains at the origin. Wow. So very important to know that. And the triglyceride content is 90. You see, very high, okay. That makes this one less dense. And then the apolipoproteins, I'll talk about them in no time, that are bound to this chylomicrons. They are the ApoA, ApoB, 48C and E, okay. So if you take VLDL, the triglyceride content is 60. Then the apo bound to it are ApoB 100, ApoC and ApoE. E mobility, we have pre-beta, okay. So the rest triglyceride content, IDL 30, 5 and so that's the table talking about. Very important. So proteins, we talk about the apolipoprotein. So now we have established the meaning of lipoproteins, their role and their function. Remember that I said the lipoproteins, okay, they transport lipids in the plasma, in the aqueous region of the plasma. And of course the lipoproteins, they have, if you look at the aspect C model, the inner core consists of triacylglyceride and cholesterol ester, which is hydrophobic, but the outer core, which consists of free cholesterol and phospholipids, and the apolipoproteins, are hydrophilic, and so they appear to solve the problem of transporting lipids in plasma, okay. Very important. And I've given you some common lipoproteins and the apolipoproteins that are bound, okay. So let's talk about the apolipoproteins. We say that they are proteins which are found in association with plasma lipids. Very important. So most apolipoproteins can be transferred from one lipoprotein to another, except ApoB, which remains with the lipoproteins in which it was initially assembled and secreted into, okay. So there's exchange of lipoproteins. When we get there, I will talk more about that. Now apoproteins bind to lipids are called lipoproteins, okay. So when you have lipids that are bound to apolipoprotein, we call them lipoproteins, okay. So these are some, this table is very important, okay. Some apolipoproteins, their site of synthesis and where they can be found, the type of lipoprotein they are bound to, and their functions. So if you take ApoA1, let me start from ApoA1. This one, A, liver, but my interest is in ApoA1. Can be found, or it can be synthesized in the GIT, the liver, which is mostly found on chylomicron and HDL, high density lipoprotein. Very important. So we found some content of ApoA1 on chylomicron and high density lipoprotein. What is its function? Most of the time, it's a structure protein of LPL, lipoprotein lipase. Lipoprotein lipase. It's an enzyme, okay, which breaks down lipoprotein in the aqueous region of the blood. That's the plasma. So this ApoA1 serves as a structure protein. Another function is that is an activator of EDCAT. EDCAT basically is an enzyme. We talk about it when we're talking about lipid transport. Okay, actually the full meaning is lecithin cholesterol ester transferase. This enzyme is very important when it comes to the transport of lipoproteins and lipids in the blood. Okay, so remember that ApoA1 can be found on chylomicron and HDL, and its function is that it's an activator of EDCAT. Okay, you come there. Then ApoA2 is also synthesized in the GIT, the liver, and then it is found in HDL. So ApoA2 is predominantly found in high density lipoprotein. It's a structure protein of HDL. Okay, very important. How we understand structure proteins? Okay, they form part of the HDL. Now ApoB48 can be found in the GIT, chylomicron, and the structure protein of chylomicron. Very important. So ApoB100 liver, that one can be found on VLDL, HDL, and LDL. They are a structure protein of them. Okay, very important. Now ApoC1 can be found in the liver, or can be synthesized in the liver, but it can also be found on chylomicron and VLDL. It's like the ApoA1. Okay, look at that. ApoA1 can be found on chylomicron and HDL, but the C1 is chylomicron and VLDL. Take note of that. Very, very important. So here it serves as an activator of EDCAT, and it modulates the clearance of VLDL from plasma. Okay, from plasma. We talk about them more in today. So when you're looking at transport of lipoproteins. Then ApoC2 predominantly synthesized in the liver. That one is found on chylomicron, VLDL, HDL. It is an also activator of EDCAT. Okay. Then the last one is ApoC3 liver. These are the lipoproteins that can be located with, and it is inhibitor of lipoprotein lipase. It means that people who lack the ApoC3 cannot inhibit lipoprotein lipase, and it may be a problem. Okay, very important. So lipid transport and metabolism in plasma. So during circulation in plasma, there are a number of processes that take place which change their composition, changes the shape and size of the originally secreted lipoproteins. Okay, so if it's like we have VLDL in circulation, okay, a number of processes occur, and at the end of the process, the shape, the size, and composition of this VLDL may change. Okay, and this may result in formation of new lipoproteins, as we talk about. Okay, some of these processes are the transfer of apolipoproteins between lipoproteins. So for instance, if you look at the exogenous pathway, there is transfer of lipoproteins between chylomicron and HDL, okay, which results in formation of different apolipoproteins. So very important. And there's transfer of lipid between lipoproteins, okay, and there's metabolism of lipid content by enzymatic action. For example, there's hydrolysis of triacylglyceride by lipoprotein lipase, this enzyme, and hepatic triglyceride lipase. These two enzymes are very important, okay, when it comes to metabolism of lipid content. So we talk about them into details when we are discussing the exogenous pathway. And after the hydrolysis of triacylglyceride by lipoprotein lipase or the hepatic triglyceride lipase, there is an esterification of free cholesterol. Okay, there's an esterification of free cholesterol by lecithin cholesterol ether transferase. So we talk about why there is an esterification. Okay, very important. And then there's clearance of lipoproteins from plasma via receptor, mainly by the liver. Okay, so lipoproteins might be cleared from the plasma and it's through some receptors in the liver. Now this operation of this process can be conceptualized as three interconnected circuits or pathways. Okay, so all these processes that I'm talking about can be grouped into three pathways, and then these pathways are the exogenous pathway, the endogenous pathway, and then reverse cholesterol transport pathway. So I'll end this video here. My next video I'll only talk about the exogenous pathway, then I'll take another video on the endogenous pathway, and then the last one. Very important. Try to subscribe to the channel for more of these videos.